Hey y'all, I'm here to talk about the complement system or the complement cascade. It's a pretty complicated system. I've always struggled with it, but I did a deep dive, learned a few things, and I figured I could try and help y'all out with it too. So let's go. There's three pathways. It's the classical, the alternative, and the Mannose-binding Lectin pathway. And first we're gonna go over the classical, right? I'm gonna talk about my materials, which is what I'm holding right here. These random deck of farm flashcards are going to be your IgM antibodies. They're crucial for the initial stages of infection. This is going to be your C2 complement protein, your C4 complement protein, C1, which binds to the IgM, we'll see soon, and this is going to be my C3. IgM produced via B cells, all right, B lymphocytes. In the initial stages of infection, as I stated earlier, they're going to bind to the pathogen surface. IgM is pretty big. It doesn't usually move through the tissue, so it's going to be floating in the bloodstream. So it's going to bind to the pathogens in the bloodstream. And then C1 is going to then bind to IgM antibody. This C1, once it binds to the IgM, it's activated. Now it's going to be able to cleave molecules, all right? So now comes in your C4 and your C2. They bind to your C1, and C1 cleaves C2 into C2b, C2a. You're going to discard C2b, and it cleaves C4 into C4a and C4b. You're going to discard C4a. So what you have now is C2A and C4B combining, forming a C3 convertase, which means it cleaves the C3 molecules into two pieces, as the C1 just did to both of those. So you're gonna put away the C1 because these are going to detach from that, free floating. It's going to find a C3, it's going to bind to that, and it's going to split up the C3 into two pieces. So you have C3A, which is an anaphylatoxin. It mediates histamine release and increased vascular permeability, vasodilation, leukocyte extravasation. We'll go over a little bit more later. And then you have C3B. Its function, or C3b's function, is to mediate CR1 Phagocytosis and the membrane attack complex using complement protein C5b through C9. I'll explain the membrane attack complex a little bit later, but now let's go over the alternative pathway. Okay, so this is the alternative pathway, and for this one, these highlighters are both going to be representing factor B. This is C3, and this is C3 with water, H2O, also known as IC3. And this is going to be factor D. Wait, factor D. Got a C3, it's coming along, you make water, or it merges with water. Okay, so now you got C3, H2O, or IC3, right? IC3 is going to bind factor B, and then factor D comes in, binds to factor B, and cleaves it, all right? And we discard factor BA, and we're left with factor BB. And so now you have IC3, BB, uppercase B, lowercase b. This is also a C3 convertase. If you remember from the letters and numbers up here, that's also a C3 convertase in the classical pathway. But in the alternative pathway, it's going to be IC3 and factor B, lowercase b. This is a C3 convertase. So a C3 binds this, IC3BB, and then it cleaves, turning into C3A, an anaphylatoxin, and C3B. C3B, if you remember, goes through CR1 phagocytosis and membrane attack complex, MAC or C5b through C9. The only thing you need to know about CR1 phagocytosis is that it leads to phagocytosis of any opsonized microbe or pathogen. But if you want to go deeper, I'll explain it a little bit later in this video. Before we go into the Mannose-binding Lectin pathway, I want to say this. So in the alternative pathway, when C3b binds to a pathogen, it actually forms a positive feedback loop where the C3 binds, leads to phagocytosis and membrane attack complex, and then it can actually bind to factor B again, and factor D will bind and cleave it like I showed you earlier, and that process will continue to go. It'll keep cleaving C3s and keep the process going continuously. Also, if you needed to know this, the C3 convertase in the alternative pathway, C3BB, is stabilized by properdine or factor P or factor P, that's if you needed to know. Okay, and the Mannose-binding Lectin pathway is going to be similar to the classical pathway. For our materials, we're going to use this again. This is going to be our Mannose-binding Lectin or Mannose-binding protein. This is going to be C2 again. It's going to be C4, and this is going to be the MBL-associated serine protease, also known as MASP or MASP, right? The Mannose-binding lectin is going to bind to mannose on a pathogen surface, right? And then MASP, the serine proteases, are going to bind. It's going to be activated by this binding, and then C2 and C4 are going to bind. Again, you're going to cleave them, get rid of C2b, so you're left with C2a. Get rid of C4a, so you're left with C4b, C2a, and then this is going to be the C3 convertase. This will also cleave C3 to C3a and C3b. And that's it. Alright, y'all, so a lot of letters and numbers that I've been throwing around due to cleaving different molecules, right? And if you thought it was gonna get any easier, you're quite wrong, okay? The reason I say that is because the one last thing that we have to go through, it's pretty hard. I wanna go over membrane attack complex, right? The C5b through C9 complement proteins. And in order to do that, we have to go over C5 convertase. C5 convertase is dependent on whether you're talking about the classical pathway or the alternative pathway. So for the classical pathway, it would be C3b, C4b, C2a. That would be the C5 convertase. Now, if you notice, you got C3b, which is part of C3 that leads to phagocytosis as well as membrane attack complex. And C4a or C4b and C2a are C3 convertase. So the C5 convertase for the classical pathway is going to be the C3 convertase plus C3b. Okay. Now for the alternative pathway, the convertase is, or the C3 convertase is C3 factor B, right? So it's going to be two C3b's and C3b. You're gonna have the C3 convertase binding to another C3, making it C3b. And then you're going to have another C3b coming in and binding to this complex, forming a C5 convertase. Now, C5 convertase splits C5 into C5A and C5B. C5A is an anaphylatoxin, just like C3A, and it leads to histamine release, or mediates histamine release, leading to vasodilation, increased vascular permeability, you know, the works. Now with C5B, this is where everything gets interesting. So C5B is going to bind to C6, complement protein 6, as well as complement protein 7. So C7 has a hydrophobic site in which it will implant itself into the lipid bilayer. After it does that, C8 is going to join in and do the same thing, where it's going to also implant in the lipid bilayer. After that, it's going to allow C9 to bind into the lipid bilayer, and C9 actually polymerizes, forming a circular pore, and that's what the membrane attack complex is. It's a pore formed in the outer membrane of the bacteria or the microbe, and this pore messes with cellular ionic gradients. So it's not a good thing for any microbes, and that's exactly what we want. Now, CR1 phagocytosis, again, like I said, you don't really need to know anything much about this, but if you do, if you're interested, here you go. It's activated by the phospholipase D pathway, and PLD activation leads to the production of CR1 in CR1 phagocytosis and CR3 proteins. These are complement receptor proteins, and these actually lead to the mobilization of leukocytes or phagocytes, and it actually facilitates the phagocytosis of these microbes and or bacterial cells. That's all I know, so hopefully that's enough for you and your curiosity. One thing I wanted to end the video with, so Mannose-binding Lectin, or Mannose-binding protein, that's actually an acute phase reactant. And where do we see acute phase reactants? That's right, in inflammation, right? So whenever you have an acute inflammatory response, your body or your liver produces acute phase reactants in order to help fight that off. So Mannose-binding protein, once it's released by inflammation or mediated by inflammation, and it will help complement activation by binding pathogens, which could be the driver of that inflammation. Bacterial infection could be the driver of that inflammation. So releasing this Mannose-binding protein and undergoing the Mannose-binding lectin pathway would be very beneficial to the body. Just wanted to say that. But all right, y'all, you have a good day.